A new chemical probe protects healthy cells from DNA damage, preserving them from one of the 8 hallmarks of aging. The story of this potentially paradigmatic development begins where so much of human health begins, the mitochondria. These organelles are disrespectfully monikered as the powerhouses of the cell, but they do so much more than just provide cellular energy. It's so important, and it even has its own DNA. Mitochondrial DNA, mtDNA, is separate from the DNA housed in the cellular nucleus. While nuclear DNA contains the vast majority of the genetic code, mitochondria carry their own smaller genomes that are essential for cellular functions. mtDNA exists in multiple copies per cell, but when damage occurs, these copies are often degraded rather than repaired. If left unchecked, this degradation can set off a cascade of failures linked to heart condition, neurodegeneration, and chronic inflammation. Published in the German Chemical Society journal Applied Chemistry, International Edition, researchers at UC Riverside developed a chemical probe that binds to damaged sites in the mitochondrial DNA and blocks enzymatic processes that lead to its degradation. There are already pathways in cells that attempt to repair, said Linlin Zhao, UCR Associate Professor of Chemistry, who led the project. But degradation happens more frequently than repair due to the redundancy of mtDNA molecules in mitochondria. Our strategy is to stop the loss before it becomes a problem. The new molecule includes 2 key components, one that recognizes and attaches the damaged DNA, and another that ensures it is delivered specifically to mitochondria, leaving nuclear DNA unaffected. In lab tests as well as studies using living cells, the probe significantly reduced mtDNA loss after lab-induced damage mimicking exposure to toxic chemicals such as nitrosamines, which are common environmental pollutants found in processed foods, water, and cigarette smoke. In cells treated with the probe molecule, mtDNA levels remain higher, which could be critical for maintaining energy production in vulnerable tissues such as the heart and brain. Mitochondrial DNA loss is increasingly linked to a range of diseases from multi-organ mitochondrial depletion syndrome to chronic inflammatory conditions such as diabetes, Alzheimer's, arthritis, and inflammatory bowel disease. When mtDNA fragments escape from mitochondria into the rest of the cell, they can act as distress signals that activate immune responses. If we can retain the DNA inside the mitochondria, we might be able to prevent those downstream signals that cause inflammation, Zhao said. Importantly, the researchers found that the protected DNA remained functional despite being chemically tagged. We thought adding a bulky chemical might prevent the DNA from working properly, Zhao said, but to our surprise, it was still able to support transcription, the process cells use to turn DNA into RNA and then into proteins. That opens the door for therapeutic applications. The project builds on more than 2 years of research into cellular mechanisms that govern mtDNA processing. While additional studies are needed to explore clinical potential, the new molecule represents a paradigm shift. Indeed, DNA damage makes up 2 of the 8 hallmarks of aging first outlined in a landmark paper in 2013, which also includes mitochondrial dysfunction as an antagonist hallmark, i.e., a result of DNA damage. This is a chemical approach to prevention, not just repair, Zhao said. It's a new way of thinking about how to defend the genome under stress. A temporary tattoo that detects drugs and drinks could prevent spiking with roofies. The news seeker responds within 1 second to even low concentrations of the drug GHB, according to scientists. When drinking in a social setting, downing a drink spiked with drugs including GHB or rohypnol, known as roofies, is an ever-present danger. The drugs are colorless and tasteless, and induce a stupor in the victim that's similar to severe alcohol intoxication, which leaves people vulnerable to being overpowered. But people can protect themselves by testing their drinks with specialized strips or cards that change color if exposed to the drugs. However, current tests are often conspicuous or take minutes to report the results. Now researchers in South Korea have developed a rapid-acting test for GHB that's worn on the skin using a tattoo-like sticker. To create the stickers, the researchers placed a mold over top a thin plastic film decorated with tattoo-like designs. The team poured a gel mixture into the mold, containing a chemical receptor that turns red when it detects GHB. They then coated the back of the sticker with a diluted glue solution so it could stick to the skin. The researchers tested the sticker's ability to detect small amounts of GHB in a range of drinks, including whiskey, vodka, beer, and coffee. The findings published in the Journal of ACS Sensors showed that within a single second, it detected the drug across a range of concentrations and below a level that would induce serious physiological symptoms. 0.01 micrograms of GHB in 1 milliliter of a beverage. In practice, a wearer could dip a finger into the beverage, touch the drop to the sticker, and see the results almost immediately, said study co-author Dr. Jeong Choil Ko of the Korea Research Institute of Bioscience and Biotechnology in a media release. And the sticker displays its positive results for up to 30 days after detection, which could be important if it's needed as a form of evidence of tampering. The researchers say that the sticker technology, funded in part by funding from the National Research Foundation of Korea and the Ministry of Food and Drug Safety, it's cheap and easy to manufacture and could be commercially available soon. Following the powerful earthquake originating off the coast of Russia, a UN organization's early warning system triggered a tsunami alert within just 10 minutes. Thanks to this global monitoring system, which has been deployed for more than 20 years, millions of people were warned ahead of the coming danger, and if a tsunami had been on its way, they would have been given precious time to evacuate. The United Nations Educational, Scientific, and Cultural Organization, UNESCO, is mostly known for promoting heritage tourism through its famous World Heritage Site designations. But the night of July 29th, an 8.8 magnitude undersea earthquake struck off the coast of the Kamchatka Peninsula in Russia. This exceptionally powerful earthquake was the strongest recorded since the Tohoku earthquake in Japan in 2011 and one of the 10 strongest since 1900. Just 10 minutes after the earthquake, the Pacific Tsunami Warning System, coordinated with UNESCO, issued an initial warning covering the areas most at risk, particularly the Russian and Japanese coasts. This warning was then relayed by national centers and enabled the immediate implementation of evacuation plans in several countries. Within 20 minutes of the earthquake, this system provided detailed forecasts of expected flood heights, and the alert was then extended to other countries bordering the Pacific Ocean. Many of these countries, including China, Indonesia, Mexico, the Philippines, Peru, the United States, and New Zealand, then issued national alerts or carried out preventative evacuations. This timely alert once again demonstrates the crucial role of international scientific cooperation in the face of natural hazards. UNESCO oversees a global tsunami warning system, puts ocean science to work to protect millions of lives, and helps communities prepare for this risk, said Audrey Azoulay, Director General of UNESCO. It's proof that the S in UNESCO is just as important, if not more so, than any other letter in its beloved acronym. Established after the 2004 Indian Ocean tsunami, which claimed more than 220,000 lives, the Global Tsunami Warning System, coordinated by UNESCO, relies on a dense network of sensors, tide gauges, and regional warning centers. This system is based on an alert chain combining scientific expertise, international coordination, and the rapid response capacity of local authorities. First, seismographs in place for earthquake detection and preparedness relay information on the severity of the quake to regional warning stations, wherein scientists and operators plot the course of the hypothetical tsunami from the quake's epicenter and leverage the readings of 1,400 sea level monitors contributed by UNESCO member states. Little more than buoys, these buoys measure changes in the water level that indicate a large wave may have passed. That information can tell you when, where, and how big a tsunami will be, said UNESCO's head of Tsunami Resilience Section, Bernardo Aliaga. 20 years ago, Aliaga said that it might take hours to generate and gather this amount of data, which is now all done in 10 minutes. This system now covers the Pacific, Indian, Caribbean, Northeast Atlantic, and Mediterranean Ocean basins. Beyond alerting communities when tsunamis occur, UNESCO is working to strengthen the resilience of coastal population through several key initiatives. UNESCO's Tsunami Ready program, implemented in 43 countries, trains coastal communities in tsunami risk prevention through evacuation plans, information campaigns, and local warning systems. Full-scale evacuation drills are regularly organized to test the effectiveness of warning system and raise awareness among populations. In response to the potential damage caused by the tsunami, Director General Azoulay has pledged UNESCO's support to coastal communities, particularly in preserving and restoring their natural and cultural heritage. UNESCO also actively supports scientific research in this field to better understand how tsunamis occur, move, and impact coastal areas. James McCullough was in the lab extracting tiny cells called mitochondria from cells when researchers on his teams rushed in. They'd been operating on a pig heart and couldn't get it pumping normally again. McCullough studies heart damage prevention at Boston's Children's Hospital and Harvard Medical School and was keenly interested in mitochondria. These power-producing organelles are particularly important for organs like the heart that have high energy needs. McCullough had been wondering whether transplanting healthy mitochondria into injured hearts might help restore their function. The pig's heart was graying rapidly, so McCullough decided to try it. He loaded a syringe with extracted mitochondria and injected them directly into the heart. Before his eyes, it began beating normally, returning to its rosy hue. Since that day almost 20 years ago, McCullough and other researchers have replicated that success in pigs and other animals. Human transplantations followed. In babies who suffer complications from heart surgery, sparking a new field of research using mitochondrial transportation to treat damaged organs and disease. In the last 5 years, a widening array of scientists have begun exploring mitochondrial transplantation for heart damage after cardiac arrest, brain damage following stroke, and damage to organs destined for transplantation. Mitochondria are best known for producing usable energy for cells, but they also send molecular signals that help keep the body in equilibrium and manage its immune and stress responses. Some types of cells may naturally donate healthy mitochondria to other cells in need, such as brain cells after a stroke, in a process called mitochondrial transfer. So the idea that clinicians could boost this process by transplanting mitochondria to reinvigorate injured tissue made sense to some scientists. From studies in rabbit and heart cells, Macaulay's group has reported that the plasma membranes of cells engulf the mitochondria and shuttle them inside, where they fuse with the cell's internal mitochondria. There, they seem to cause molecular changes that help recover heart function, when comparing blood and oxygen-deprived pig hearts treated with mitochondria to ones receiving placebos. Macaulay's group saw differences in gene activity and proteins that indicated less cell death and less inflammation. About 10 years ago, Sitaram Imani, a cardiac surgeon at Boston's Children's Hospital, reached out to Macaulay about his work with animal hearts. Imani had seen how some babies with heart defects couldn't fully recover after heart surgery complications and wondered whether Macaulay's mitochondria transplantation method could help them. During surgery to repair heart defects, surgeons use a drug to stop the heart so they can operate. But if the heart is deprived of blood and oxygen for too long, mitochondria starts to fail and cells start to die, in a condition called ischemia. When blood begins flowing again, instead of returning the heart to its normal state, it can damage and kill more cells, resulting in ischemia-reperfusion injury. Since Macaulay's 8 years of studies in rabbits and pigs haven't revealed safety concerns with mitochondria transplantation, Macaulay and Manny thought it would be worth trying in procedures in babies unlikely to gain heart function. Parents of 10 patients agreed to the experimental procedure, which was approved by the Institute's review board. In a pilot that ran from 2015 to 2018, Macaulay extracted pencil eraser-sized muscle samples from the incision made for the heart surgery, used a filtration technique and isolated mitochondria, and checked that they were functional. Then the team injected the organelles into the baby's heart. 8 of those 10 babies regained enough heart function to come off life support, compared to just 4 out of 14 similar cases from 2002 to 2018 that were used for historical comparison. The treatment also shortened recovery time, which averaged 2 days in the mitochondrial transplant group compared with 9 days in the historical control group. 2 patients did not survive. In one case, the intervention came after the rest of the baby's organs began failing. In another, a lung issue developed 4 months later. The group has now performed this procedure on 17 babies. The transplant procedure remains experimental and is not yet practical for wider clinical use, but Macaulay hopes that it can one day be used to treat kidney, lung, liver, and limb injuries from interrupted blood flow. It will take more research into the mechanisms of mitochondrial therapy, improved mitochondrial delivery techniques, larger trials, and a body of reported success before mitochondrial transplants can be FDA approved and broadly used to treat ischemia-reperfusion injuries, researchers say. The ultimate goal will be to create a universal supply of stored mitochondria, a mitochondria bank of sorts that can be tapped for transplantations by a variety of healthcare providers. A groundbreaking new material made from upcycled rice husks in a zero-waste environment is giving home builders the option to reduce their carbon footprint while adding a rustic charm to their home's exterior. The product, called Acre, offers the warmth and beauty of real wood with all the conveniences of composites and is available as siding, decking, fencing, sheets, and trimming. If any Americans were following the news during trade negotiations between Japan and the US, they may have learned, and maybe were surprised to do so, that US farmers grow 20 billion pounds of rice every year. Each of these pounds are made up of hundreds of individual rice grains, each of which is covered by a hard husk, sometimes called a hull, which are normally just sent to the landfill. Acre inventor and producer Modern Mill is changing that by transforming this overlooked byproduct into a high-performance wood replacement that doesn't require cutting down a single tree, and they've already spared 4,000 tons of rice husks from the landfill. Unlike wheat, the rice hulls contain a significant amount of lignin, the organic polymer that forms wood. This makes them strong and durable when processed, and capable of being harnessed for use as a building material. The husks themselves offer a tone and depth of color that the company describes as imitating tropical hardwoods like teak, ipe, and cedar. 1 pallet of Acre material, therefore, saves a whole acre of tropical rainforest hardwoods, and in 2022 alone, 44,000 of these pallets were delivered to builders around the country. Though prices can vary significantly, most building projects their siding will cost between $10 and $12 per square foot. In 2023, Modern Mill was named to Fast Company magazine's Most Innovative Companies list, and in 2025, in response to devastating Palisades fires in California, Modern Mill donates sidings to 20 families who lost their homes to aid recovery and rebuilding efforts. Our hearts are with the families and communities affected by the wildfires, said Chris Guyman, Chief Executive Officer at Modern Mill. We're doing everything we can to support relief efforts. Acre is tested and certified in accordance with California Fire Code to be used in all areas from moderate to very high fire hazard severity. 2 cities in the Netherlands are mounting tiny staircases on the sides of their canals to help cats escape if they fall in the water. Between the capital of Amsterdam and a smaller city called Amersfoort, there could be over 500 tiny staircases along the canals by the end of the year. The initiative began when animal welfare group Party for the Animals noted a high number of drowned felines in the Amsterdam Canals network. Locating a fund worth €100,000 that was currently unutilized in the city's environmental and biodiversity budget, the party leader Judith Krom proposed that it go to funding exit points for cats to use for escaping the canals. Animal rights councillor Zita Pels supported the plan, and on July 10th, the Amsterdam City Council voted in favor of Krom's proposal. A simple measure can prevent enormous animal suffering, Krom said. The adopted motion demonstrates that as a city, we take responsibility for protecting the lives of animals. The current plan is for the government to work with Die Ren Ambulance Amsterdam, another animal welfare organization, to identify areas where cats fall into the canals the most and build the staircases there. Amersfoort, 31 miles southeast of Amsterdam, is probably a much better place to go and see tiny kitty staircases. The municipality currently intends to build hundreds every year of these life-saving features. For 10 million worldwide patients with Parkinson's disease, news out of Japan that a large pharmaceutical corporation is seeking approval for a new stem cell-based treatment should be hugely encouraging. Following successful clinical trial in which seniors between 50 and 59 years of age received injections of induced pluripotent stem cells, IPS, and saw either a halting or reversal in symptoms, Sumitomo Pharma have applied for manufacturing and marketing authorizations in Japan and the United States. Parkinson's is a debilitating neurodegenerative disease characterized by a progressive loss of dopamine-producing cells that results in a loss of motor functions. The clinical trial led by Kyoto University and the published results in Nature showed that 4 out of 7 patients saw improved symptoms during a 2-year monitoring period, while the other 3 did not, but suffered no negative side effects. Induced pluripotent stem cells are cells in the body that have been reprogrammed to return to a very young state, and which can then form any cell in the body. The discovery of the genetic process was made in part by Dr. Shinya Yamanaka and the Kyoto University research team. Together, they won the Nobel Prize for this work. It allowed stem cell treatment research to completely bypass the existing ethical debate around the harvesting and use of placental or fetal stem cells. The 4 so-called Yamanaka factors are genes which, when altered, cause a cell to return to a youthful state. In the clinical trial, IPS from healthy donors were programmed to form dopamine-producing cells lost in Parkinson's cases and given in the form of 2 shots, one on each side of the brain. Currently available therapies improve symptoms without slowing or halting disease progression, says the Parkinson's Disease Foundation. Famous sufferers of Parkinson's disease include actor Michael J. Fox, boxer Muhammad Ali, and singer Ozzy Osbourne, who recently died last month. If you like more daily doses of positive news, please subscribe to my channel. Please also like and leave a comment, as this really helps the channel grow. Links to the article on this news story can be found in the description of this episode. Thank you for watching.